Hey guys, welcome to Chapter 9. This is the first chapter in Unit 3, and it's one of the three chapters that I think is the most important for you as pre-healthcare majors to be paying attention to in this class. And I really feel that these are the topics of why the nursing program and other healthcare programs are asking you to take this class. So it's really important that you pay attention to the details in these next three topics because I will be asking you specifics about them. Usually we talk about broad concepts, but here we need to know the details. So in topic 1, we're going to talk about the overview of controlling microbes. So why is this topic important? In a hospital or a clinical setting, the main way that we prevent infection is by controlling microbial growth. So it's really important that you understand the basics of this so we can talk about the two main ways, chemical and physical controls, in the next two topics. Here are our topic objectives. These are fairly consistent, but there's a few terms you need to know as well as some big concepts I want you to be able to understand. So it's a combination of very specific information and a very general information. So make sure you understand the two of these. So first of all, let's start with possible outcomes of microbial control. There are four main possible outcomes that I want you to be familiar with. The first of these is sterilization. Now this is the gold standard. This is what we'd love to be able to accomplish in all situations, but it's just not possible. And this is where we destroy and remove all viable microorganisms. This includes viruses, it's everything. Now, the reason why we can't do this for everything is because these processes are so intense that they would potentially kill us or at least really damage our cells in the process. Or the material that we're trying to sanitize. So not everything can be sterilized. So that leads us to three other types of outcomes for microbial control. We have disinfection, and this is where we use a chemical agent to remove vegetative cells from some kind of inanimate object. We can usually also remove microorganisms or toxins from them or anything else, but the big thing is that usually spores are not captured through a disinfection process. So when you're wiping something down with bleach, this is an example of disinfection. Another example is decontamination or sanitation. This is usually some kind of mechanical process, either a filtration or the use of soaps or detergents to knock down the quantity of microbes on a surface. So it's a little bit lower level, but it still does help. In this case, anything helps. So if you can keep all the microbes at a safe level, then we don't have to worry too much about the spread of infection. Then the last one is the one that actually is the only one that we can apply to ourselves. And these are antiseptics or degermination. And this is where we use detergents, hand scrubs, alcohol, etc. on our skin and other surfaces to reduce the number of microbes present. Remember, we don't want to kill them all because we do want to keep some of our natural flora. But the key is to keep them at bay so we don't develop any opportunistic infections through a microbe that might already be present on the skin. So what you can see here on this graph is the four different types of microbial control using a variety of different methods to get there. And we're going to talk about these methods in the next two topic lectures when we talk about physical agents and chemical agents. And we group mechanical agents in with physical agents, but we'll talk about that when we get to physical agents. But as you can see here, the only place where antisepsis works is on animate objects. You, me, your dog, your cat, things like that. That's antisepsis. And decontamination only happens here in air. The other one, sterilization, disinfecting, can happen in a variety of places, but this is always on inanimate objects. So this never happens on living tissue. So pay attention to this chart. This chart's a really good place to reference back to for the next couple topics because it'll really play a role in everything else. So how do these methods work on microbes? That's a big question, right? And so what you can see here is a listing from more resistant to less resistant microbes. And we've talked about this a lot. We've talked about how spores are really hard to kill. And prions are actually even harder to kill, but we never really talked too much about prions because they're not something that's easily communicable, so we don't worry too much about them. Whereas bacterial endospores, mycobacterium, and staphylococcus, things like that, are much more resistant to methods than, for instance, gram-positive bacteria. So I want you to be aware of this chart and how it relates to things. And then take a look over here at the other chart, and you can see where it's comparing how much more resistant endospores are than vegetative cells to a variety of different a variety of different methodologies. So pay attention to that. And the big thing I want you to remember from here is really kind of where things fall. The spores, the cysts, they're going to be more resistant than the gram-positive bacteria and some of the viruses in general to a variety of methods. So we have a couple important terms that we need to get straight to it before we can go into the next two topics. And this is agents and processes. So agents are the tools that we use to do something. That something we're going to do is a process. So for example, if I want to sterilize some surgical tools, that is a process. Sterilization is my process. Now, if I'm going to use x-rays to do that, I would use the x-rays in order to sterilize, so that's my agent. The same thing comes with bleach. If I'm disinfecting something, disinfection is my process, whereas bleach is my agent. A couple other important terms to get under control before we move on are anything that ends with a "cide." So bactericide, fungicide, viruside, these are agents that can kill the microbes. They've been tested to be able to kill them. Static on the other side, so bacterial static, fungistatic, virostatic. This means it prevents further growth. So it doesn't necessarily knock down the volume of cells or anything else, but it does inhibit growth. One last set of terms, and these ones are something you might be familiar with, but sepsis is what happens when microbes get in the blood. This is a huge deal, right? If someone gets a bacterial infection that spreads the blood, then it's really serious. And so this is known as sepsis. The way we prevent that is through asepsis. That's a process. And then the agent we use is antiseptics. And these are chemicals used for asepsis. So be familiar with these because you'll see these terms a lot in your future careers. So practical matters. If you were asked to go clean a room, because somebody was just in there that had tuberculosis, for instance. There's a lot of different questions you have to ask. Now, granted, if you're in a hospital, there'll be standard operating procedures for you to use, but there's some important questions that go into these. So you can see here, the first of them is, what do we need to do? Do we need to sterilize, or can we just disinfect? For instance, in the laboratory, we just disinfect. We don't ever sterilize the lab benches, for instance. And that's because disinfection is adequate for this situation. Another thing to consider is, can we keep this item or can be discarded? So for instance, we will sterilize all the autoclavable waste that leaves that lab. That's why we have that red bin in the lab. And so that is permanently discarded, so it's okay for us to sterilize it at really hot temperatures in the autoclave. And so if it's going to be reused, then we have to talk about what kind of methods of disinfection we can use. Can it withstand heat, pressure, radiation? Usually at laboratory or hospital type tools are graded for specific things, especially heat, so they can be autoclaved. And then you have is the control method appropriate for the application? Does it work? Is it going to leave a residue if we're using chemicals? Is the agent able to actually do the job? And lastly, and it's always a matter with anything in the lab or in a hospital, is it cost effective? If you were to have to boil water to scrub everything down, that's going to be really time intensive and not very effective for labor use. And so that's why, it might be better to get some spray that is able to do the same thing. And so those are some of the practical matters that come into it. So I want you to really be familiar with these questions and be able to analyze them and see and determine how they're important to the selection of your antimicrobial mechanisms. So the last thing, and this whole topic comes right back to this, is microbial death. Our whole goal is to kill the microbes. Now, it's pretty easy to tell if you and I are alive or dead. It's pretty easy to tell if other complex organisms are alive or dead. In microbes, it's not quite as easy. So what we use as our standard for microbial death is if you take a sample from either the broth, the table, whatever, put it in some nutrient broth, nutrient agar, some kind of growth medium, and it grows and replicates, then that organism is not dead. But if it doesn't, then the broth or the station or whatever it is has been sanitized. Sterilized actually. It's been sterilized because it has killed the microbes. And so that's how we determine if a microbe is dead. So that's really important. Now there's a number of factors that affect microbial death. And these are kind of obvious factors. I mean, you look at the list and, oh my gosh, all these things I have to learn. But just think about it. If you were to spill a big old 1-liter flask of E. coli on the floor, would you take one spray of bleach and walk away? No, you'd have to use a greater amount of bleach to match it. And so that's where these factors come from. The nature of the microbes. Are you dealing with something that is really resistant, or are you dealing with something that's fairly not? As we saw from that earlier chart, if you have a gram-positive spill, the cleanup is going to be a lot less needed, or you're going to need a lot less of what you're working with, or a little bit more of a weaker chemical if you want to be cost efficient to kill that, versus if you have a staphylococcus spill. Then the temperature, pH of the environment, as we've learned, that those can change things. So if it's a really extreme temperature, you're not going to have to worry as much. The same thing then comes in for the agent. If you're using full out bleach, 100% mixture, you don't need very much. A little bit goes a long way. Whereas if you're using a 1 to 100 dilution, you're going to need a lot more to completely kill the microbes. So that's where these things kind of come into play. So be able to understand what these factors are and how they play a role in the decontamination microbial control process. So the last thing before we move on to our next set of topics on actual controls is looking at how antimicrobials work. Now, I fully expect you to have these 4 targets and examples of them and how they work under control. I expect you to have these practically memorized. This is really important. There are 4 main mechanisms that we act on. And we talk about proteins a lot because moist heat is one of the ways that we kill microbes, and it's the easiest way. Most people have an autoclave or something like that that they can destroy things with. And what happens with proteins is we just denature the proteins. We cause them to unfold. Proteins don't refold naturally, and so it's an easy way to kill things. Now, one of the things that's really important when we focus on these is we want to make sure that we're using something that can target these cells, but if it's going to come into contact with humans, we need to make sure it targets only prokaryotic or viral or whatever our target is, targets something that is different from them versus us. For instance, in the cell membrane, if it's going to be on a surface that we may be touching, you don't want it to target the phospholipids that we have in our membrane just as well as Staphylococcus has in its. Instead, we want to target a specific Staphylococcus protein so that it doesn't harm us. Now, that doesn't always happen. If you look at x-rays, heat, etc., those things are just as damaging to us. And so that's why we have to take protective action when we're using those things. For instance, we're going to use x-rays, we stay out of the room when we're using them. Or if we're using the autoclave, it's in this really packaged heat wrap container to protect us from that heat and the moisture. So really spend some time with this, and if you have any questions about it, let me know. And this is the end of our topic on introducing microbial controls. So let me know if you have any questions about these. As I said, I expect you to really have a lot of these specifics. And I know that it probably feels pretty overwhelming, but I want you to make sure you have a good feel for this before you move on. So if you have any questions, as always, just email me or let me know in class. Thanks. Bye.